So I'm going to first show you a conjugal cross between an F plus and an F minus cell. Again, I've designed animations to really make this a little bit more understandable. You have a similar cartoon depiction of this in your book. So if you're looking at your book, you're going to have basically these similar figures, but they're clearly not going to move off the page. So what I'm going to do is I'm going to help them move off the page for you. Okay? So this is a conjugal cross between an F plus and an F minus. So one of the first things that has to happen is we have to make the sex pili. Remember, that's going to allow for the transfer of that fertility factor from the F plus to the F minus cell. As the plasmid replicates, remember, plasmids are going to contain information that is not essential, but the bacteria really kind of likes. It gives it some additional protection maybe in exposure to antibiotics, might make it a superbug because it renders it resistant to certain antibiotics. So the F plus cell doesn't just want to give the F minus cell its plasmid, it wants to keep a copy for itself. So what you're going to see during conjugation is as the plasmid replicates, it's going to keep a copy of the plasmid and donate a copy to the F minus cell. So here's how this goes. The plasmid is replicating. We're going to donate one copy to the F minus, and we're going to keep the original copy. What you may not have seen down at the bottom is now this cell has become F plus. That's a very important point to know about the conjugal cross between an F plus and an F minus cell. All of the recipients become F plus. All right? So you can see if, for example, there was antibiotic resistance encoded on that plasmid, it could very quickly spread through a population of bacteria, because if you add one F plus cell to a population of several 100,000 F minus cells, within a very short period of time, all of those cells in that culture will become F plus. And the reason I'm kind of making a big deal about that is because when I show you the next cross, that's not the case. So in a conjugal cross between an F plus and an F minus cell, the cells, the recipients, become F plus. Okay, now we're going to talk about the HFR transfer. The HFR, remember, is when you have a plasmid inserted into the bacterial chromosome. It still has the ability to encode for the sex pili and to allow for the transfer of DNA because it has an ORI-T. But what's the main difference? Now we're not talking about transferring a small plasmid. So if we were playing with, let's say, a rope or a string in a cross between an F plus and an F minus, I would donate to you this short piece of string. You could easily take it across that little tunnel or the sex pili. Transfer of the entire genome is like trying to transfer a ball of yarn from one bacteria to another. So it keeps going. There's a lot of DNA. And you know what happens is when those bacteria are together, bridged again by that sex pilus, there's no way they can stay together long enough to transfer the entire chromosome from point A or the HFR strain to the recipient. That would take approximately 2 hours to do that, and the cells don't stay together long enough. So you want to assume that 20 genes or less were transferred. That's about the time that you have. Now, what else is important is the transfer is unilateral, and here's the biggie. The recipient, the F minus cell, stays F minus. It does not become HFR positive. It does not become F plus. Okay. Now, since we're trying to donate this large piece of DNA, when the bacteria actually shear apart, you're going to be left with, in the recipient, a linear piece of DNA that has to be stabilized in the chromosome to be expressed, and that's done again by homologous recombination. So here's an example of HFR conjugation going from this high-frequency recombinant to an F- cell. So again, we're starting. We got the sex pili. We're going to direct transfer, but not the entire piece is going to go, only a small piece. So we're going to find an exchange. We're going to find alleles that are similar and get an exchange of genetic information, okay, rather than the addition of an entire plasmid. So an HFR conjugal cross requires homologous recombination to get expression of that linear DNA that came in. So that's an important point. And if you look at the bottom, this cell remains F-, okay? So just to summarize again for you, in an F+ to an F- cross, we're donating the whole plasmid. The recipient F- cell then becomes F+. In an HFR cross, like we just showed, the HFR has a plasmid integrated into the chromosome. It's going to start to direct the transfer, but all of those genes aren't going to make it across. And in order to get stabilized expression of that DNA, homologous recombination has to occur. Now, one of the reasons that the recipient cell stays F- is because once you begin the transfer of the chromosome, the fertility factor is one of the last to go. So it's never really going to have time, because that's at the end, to get across and make that cell HFR positive or F+. Okay? The final mechanism of gene transfer we're going to talk about is transduction. And this one gets a little bit complicated, but it's a nice little introduction to the virology chapter. Transduction deals with bacteriophage. So again, viruses that specifically infect bacteria. There are two types of transduction, and I'm going to define them for you here, and it may not make a lot of sense if you don't understand the process, but as we go through the process of transduction, I'll show you how these different types of transduction occur. Okay? We're going to be talking about two different types of phage. We're going to be talking about a lytic phage and then what we call a temperate phage. Lytic phage can only get into a bacteriophage, replicate, and blow that cell up and go on and infect another bacterial cell. Temperate phage can do that. They can do what I mentioned for the lytic phage, but they can also integrate into the chromosome. Okay, and I'm going to show you how that works. But look at these different types of transduction. You've got generalized and specialized. In generalized transduction, this is basically when all genes have an equal opportunity to be transduced. It is a packaging error of the phage that when it's trying to put all of its pieces together to make an intact phage, it accidentally grabs bacterial DNA instead of phage DNA. Now, because that's just a random event, it's an error in packaging, generalized transduction can happen in lytic infections and temperate infections, whereas specialized transduction occurs from an excision error when that temperate phage is trying to excise from the bacterial chromosome. Okay? Therefore, only temperate phage can undergo specialized transduction. All right, so let's start looking at the phage. Again, I mentioned there were two different types of phage. You have a virulent phage that blows up cells, very similar to the poliovirus. And you have a temperate phage. Temperate phage are the ones that kind of can do both. They can be lytic, or they can insert into the bacterial chromosome. Therefore, that gives us the two different options for transduction. In the lytic infection, we're going to bind to the bacterial cell. We're going to replicate much like mammalian viruses do, and we're going to blow up the cell and go on and infect another cell. Okay, so that's the essence of a lytic infection. So I'm going to show you an example first of the lytic cycle, and then I'm going to show you how generalized transduction works. Okay, so here's the lytic cycle. Again, I've animated it to help you understand how the process works. Here's your bacterium. Here's your chromosome. Your little phage comes in, and it attaches to the bacterial cell. And phage, being a little bit different from mammalian viruses that infect mammalian cells, just injects the DNA through this little tail tube into the bacterial cell. So we're going to inject that DNA into the bacterial cell, and now what this phage is going to do is going to replicate. And as it's replicating, it's going to get transcribed and translated. We're going to make proteins. And the cell now is going to start to die because it's utilizing all of its host cell machinery to replicate phage. So in a sense, it's going to start chopping up its own DNA. It's kind of ready to die. And at the same time, the phage is trying to assemble. So we have all these phage parts. We have phage DNA. We've got the tail fibers. We've got the head. Well, here's a tail fiber. Here's a tube. And here is a chopped up piece of bacterial DNA. Now, the cells, the phage, actually start to assemble. And this phage did it right. This phage did it right. This phage did it right. Oops. This red one over here took bacterial DNA. Okay? So this little red guy is the one that we're going to look at specifically for generalized transduction. It mistakenly packaged bacterial DNA when it was assembling. Therefore, when I talk about generalized transduction, so you understand how this works, any of those genes it could have possibly taken. There's no one gene that's going to be acquired more than any other. So each and every gene has an equal opportunity to be transduced by generalized transduction. So now we have our phage that are blue. Those are the ones that assembled correctly. Our red phage here is going to show us how generalized transduction works. In generalized transduction, this little guy comes in, and he has all of the attachment proteins needed to what? To bind to another bacterial cell. It's still capable of delivering genetic information from the phage to the bacteria. So it will do that. But now it didn't inject phage DNA. It injected DNA it randomly picked up from another bacterial cell. So now what's going to happen is that phage, there's no phage infection here. All it can do is attach to the cell, inject the DNA, and that's it for the phage. So what it did in this instance is it acted as a vehicle to take phage or to take bacterial chromosomal DNA from one cell to another. So now what has to happen? We basically right there have a linear piece of DNA. If this bacterial cell wants to keep it, wants to express it, what has to happen? That's right, homologous recombination. So this piece of bacterial DNA has to find a region of homology on the new bacterial chromosome and exchange the DNA. So now we have new bacterial DNA that has been exchanged with chromosomal DNA from this particular bacteria, and then this DNA, this genetic information, is lost. Okay? So that shows the lytic phase, and it shows generalized transduction. So now you can see how any gene has the possibility of being transduced by generalized transduction. Now, let's look at specialized transduction and lysogeny. Remember, temperate phage functions similar to HIV, where they can insert their DNA into the bacterial chromosome. The whole key to understanding this, though, is that these temperate phage have preferred sites of integration. They don't integrate everywhere or anywhere in the bacterial chromosome. They have preferred sites of integration. That's key to understanding the process we're about to talk about. When the repressor is made, all right, a temperate phage is going to insert the DNA into the chromosome, and it's going to keep it there. So let's look at that real quick. Again, I have just a little animation to help make this more clear. You've got phage DNA here. You've got your repressor. Here's your preferred integration site for this particular phage between the gal and bio genes, let's say of E. coli. And now we're going to integrate phage DNA into the chromosome, keeping this repressor. As long as the repressor is there, every time E. coli replicates, the phage DNA is going to replicate with it. Okay? So it's going to stay integrated into that E. coli chromosomal DNA as long as that little pink triangle, which is representing the repressor, is there. All right, now let's look at what's going to happen with specialized transduction. Lysogeny, again, the phage DNA is going to replicate with the chromosome as long as the repressor is there. Now, everybody's got to have a backup plan, right? So the phage is no different. Even being a lowly virus that doesn't even get to infect mammals, we're talking a virus that infects bacteria, it's still pretty smart, because when things get rough, when the situation gets tough, and it looks like there are maybe some chemical agents around that may kill the bacterial cell, the phage can get out. It's got a backup plan and it's ready to go. When the repressor gets damaged, now that phage is going to excise from the bacterial chromosome, and it can then go infect another bacterial cell, maybe that is not under some physiologic stressor. Okay, now we've mentioned this a few times when we've talked virulence factors in Chapter 2, but here's the nice list for you of virulence factors that were acquired through this specific mechanism that we're talking about of lysogeny. The O antigens of Salmonella, the botulism toxin, the exotoxins from Streptococcus pyogenes, remember, responsible, for example, for the fever and rash from scarlet fever, and diphtheria toxin. A few others, cholera toxin and Shiga toxin as well, were all acquired by this particular mechanism of transduction. So let's take a look. The induction. We already showed what happens when we integrate into the bacterial chromosome, again, at a specific site in the chromosome. As long as the repressor is there, again, every time the E. coli replicates, that phage DNA is going to get replicated. But now let's look at induction. If the repressor gets damaged by UV light, by cold, by other chemical agents, the repressor gets damaged and the phage excises from the bacterial chromosome. It wants out. It knows that the ship is going down and it's getting off before it sinks. Okay? So it's going to excise from the bacterial chromosome, and most of the time it does that very easily. It knows generally where to excise, where it integrated in the first place, and under normal circumstances you get a normal excision. And now this phage DNA is going to enter the lytic cycle. So we're going to start to replicate it, we're going to make phage, and now this phage can go on and infect another bacterial cell. So that's the normal process of induction. Let's look at what happens with an excision error, which is the prequel to specialized transduction. You still have some stressor, whether it's cold temperatures or UV light. The repressor gets damaged. But now you have an excision error. You don't cut exactly the phage from the bacterial chromosome. And you mistakenly take some of the bacterial chromosomal DNA with you. So now, when you go into the lytic cycle and you make phage, you have a good bit of phage DNA, but you also have a piece of bacterial DNA. This is going to allow for specialized transduction. Now, why did we say in specialized transduction only certain genes have the ability to go? Because phage integrate at certain sites. So when you get an excision error, it's more likely to be some genes over others. All right, so that's really kind of the complete story there for specialized transduction. How does it work? We get a phage, there's the excision error, and now we've taken some bacterial DNA from one cell to another. We're going to inject that DNA into the new bacteria, and guess what? We just injected bacterial DNA from one cell into another. Remember, the phage inserted between gal and bio. Okay? So now what we can do is we can take gal plus into a gal minus bacterium. How does it have to stabilize? How does it have to be expressed? Through homologous recombination. We have to find an area of homology on the bacterial chromosome, exchange it with the linear DNA, and then those alleles that were originally on the chromosome are lost. That is specialized transduction. So let me compare the three mechanisms of gene transfer that we've talked about. All right? Transformation, conjugation, transduction. Which ones require cell-cell contact? Of course, that's specific for conjugation. Which one are dependent on phage? Of course, that's transduction. Is competency required? Remember, that's specific for transformation, where the cell becomes physiologically competent to take up DNA. So that's specific for transformation. Is naked or free DNA involved? That's only relevant for transformation. Is homologous recombination required? Yes, for every transfer except an F plus to an F minus. Now, the simple summary here for generalized versus specialized transduction is, again, with generalized, you have an error in assembly, and any gene can be transferred. And with specialized, it's an excision error, and only certain genes next to the phage insertional site can be transferred.